Oh dear, where are we? Okay, so looking at proteins today, just a little basic quick one on proteins. So let's get into proteins. Okay, so proteins are the third biological molecule we're looking at in the series so far of lessons. We've already looked at carbohydrates and lipids, so we're just going to do a basic intro on proteins. It shouldn't take too long. You might remember from GCSE that proteins are made up of monomers. Can you remember what the monomers are called? What are the four? Oh my gosh, it's getting my head together. What are the four main elements from the periodic table that you would find in the monomers of proteins? For instance, in carbohydrates and lipids, we find carbon, hydrogen, and oxygen. But there's four elements that we find in proteins, sometimes five. Can you give any examples of proteins that you know? So, oh my gosh, sorry, just sorting out my, okie dokie, sorting out my connection. Right, so let's go through some of these. Hopefully you can come up with some of these in your head. If you want, you can pause the video if you're watching it on playback and test yourself on these questions. I'm about to give you the answers. So the monomers of proteins are called amino acids. The four main elements that are found in all proteins and all amino acids are carbon, hydrogen, oxygen, and nitrogen. However, there's a little bonus. Some have sulfur as well in them as well. Certain proteins have sulfur in cysteine groups. Where in the cell are proteins made? You might remember which organelle or which cell structure makes proteins. Is it the cytoplasm? Is it the cell membrane? Is it the nucleus? What actually constructs the proteins? You'll remember from GCSE, it's the ribosome or ribosome, however you like to say it. It doesn't matter. You're not going to be tested on your audio verbal skills. So can you give any examples of proteins that you learn about in GCSE? Well, some of the proteins you learn about in GCSE be things like hormones, adrenaline, testosterone, produced in the testes of men that help produce sperm and give men the secondary sexual characteristics. You've got estrogen produced in the ovaries and progesterone in the ovaries. You've got LH and FSH, you remember those, produced in the pituitary gland. And there's plenty more hormones. You've got insulin and glucagon in the control of blood glucose levels. So hormones are one type of molecule that that are made from proteins. We have enzymes. You remember your enzymes, protease, lipase, amylase. These enzymes, digestive enzymes. There's also enzymes for building things as well, which you learn more about in A-level. So enzymes are proteins. We also have antibodies. Those are proteins. You remember the antibodies, part of the immune response. B cells, B plasma cells produce antibodies that lock on to the antigen that's also made of protein or found on the cell membrane of pathogens and help destroy them. And yeah, we'll leave it at that. So there's some examples of proteins. There's plenty of other proteins you can give examples of. Hi, Karam. I don't know if, yeah, it's bio today. I don't know if you're interested in bio. You might be like, hey, I'm not really interested in bio. So the monomers of proteins, it's only a quick lesson today, shouldn't be more than 40 minutes. The monomers of proteins are amino acids. You might remember that from GCSE. And there are different amino acids, but they have a commonality in their structure. We're going to look at that now. You might remember the structure, you may not. I'm going to draw it here. Maybe I shouldn't use a big fat highlighter pen. Let's try using this pen. So again, I've made it a bit bigger than I wanted. I'll sod it. Let's just do it. Right. I better shrink that nitrogen down. That looks like the King Kong of nitrogen. That should be hanging off the Empire State's Building, that nitrogen atom. Right. Let's put that to a decent size. Okay, so all amino acids, which are the monomers, the small molecules that make up the big molecular chains of protein, all amino acids have an NH2 group known as an amine group, and glycine looks like this. They have an amine group, and then the word acid is because they contain, if you did your organic chemistry for triple science, they contain a carboxyl or carboxylic acid group or a carboxyl group. So carboxylic acid, or you can call it a carboxyl group, and an amine. So all amino acids have that. This is the simplest amino acid that there is out there in nature, and it's called glycine. And the part of it that is common with all amino acids, I'm going to circle here. This also comes up in A-level chemistry, so it's kind of like biochemistry, biological molecules. You also will be doing amino acids later on in a much later unit in chemistry as well. So you'll need to know this stuff as well if you're doing A-level chemistry. And glycine there, that part of the molecule is the same for all amino acids. Let's draw the second type of amino acid. It looks very similar. You'll notice that it has a region that is the same for all amino acids, and it's the bit I've done in green. So just bussing this out again. We've got the double bond O, carbonyl group, that's called. The carboxyl group is the COOH, or carboxylic acid group. Now, glycine has a carbon atom here, and then that carbon atom has 3 hydrogens around it like this. Okay, so notice the common structure here is in the green. And that's the part of an amino acid that's always the same, the constant part of an amino acid that's the same for all amino acids. Now, the little side chain there that's coming off that first carbon, they always have 2 carbons next to the nitrogen, but you'll see there's a little side chain coming off the first carbon, and that's the bit that's different about different amino acids. Now, there's different ways you can write it, so you can display all the bonds like this. Notice I've displayed all of the bonds. If you want, you can just write NH2 there, or H2N. And you can write CH3 here. You don't have to write a C and then display all the bonds. And over here, you can just put COOH in a line. You don't have to show all the bonds. But I'm displaying all the bonds so you can see how the amino acids are made up. Okay. All right, let's move on. So instead of writing the different structures that we can get, there's actually 20 different amino acids. Okay. So alanine and glycine are just the first 2, but there's another 18 that are found in nature in all living organisms, okay? And the only thing that changes between one monomer, one amino acid, and the next amino acid is this R group, this side chain. So alanine has an H there instead of the R, and glycine has CH3, but another amino acid might have C2H5 there, or the chain can go on and on, so there can be all different chain lengths on the different 20 amino acids. So this is how we draw the general structure of an amino acid. The only part that varies is here in what we call the R group. You remember from lipids that we can write an R group for the fatty acid chains if we like as well, because they can be different lengths and some can have double bonds. So you can shorten things in chemistry by writing an R group. So this is known as an R group. It is the part that varies for amino acids. Yes, that's absolutely right, Karam. This is the generalized version of all amino acids. There's only 20 of them that occur in nature. There's only 20 naturally. And you might, so every single protein, and there's tens of thousands of proteins out there, every single protein is made, let me shrink my head down. Every single protein is made of amino acids, and there's like 10,000 of them. Different ones, but you could make millions of different proteins. In fact, you could make trillions of different proteins, really, because there's 20. If you think about your PIN number on a bank card, there's only 4 digits, 4 different numbers, and there are millions of combinations with 4 different numbers that you're never going to guess someone's PIN. Imagine having 20 different numbers and being able to arrange them in any order you want and use any amount of each that you want. So it's just a crazy amount of different proteins we can construct by joining together different amino acids. I don't know if you know the answers to any of this, but 2 amino acids join together in a something reaction. Do you remember how we get a glycerol molecule in lipids and 3 fatty acids to join? There's a name for that reaction. And there's also a special name when we get 2 glucose molecules to join to form maltose or to form amylose or amylopectin, the different types of starch. Condensation, absolutely. And that is, again, if you're just doing chemistry, this is going to come up in chemistry as well. So condensation, it's going to come up actually in the 2nd year of chemistry, in the kind of A2 year, the year 13 stuff. You get a bit of biology coming up. Yeah, no problems. Thank you for that. Yeah, no problems at all. Cheers. If you're thinking of taking A-levels in any of the sciences, I'm going to be doing them all from September. So great stuff. Much appreciated. What the bond that forms between 2 amino acids is known as a peptide bond. This is my 3rd lesson in A-level biology, so I'm actually just going through the whole course, but when people start, they can catch up by watching these if they want in September. And I'm also going to do ones where I just take any questions, because I'm going to be doing year 13 lessons and stuff as well, so for biology and chem. So I was going to do physics as well, but it's a little bit physics is not normally as many people interested in the physics, but I might do. Okay, yeah, peptide bond, and then the molecule formed from joining 2 amino acids, actually a dipeptide, but you were spot on there, Karam. There is such a thing as, I lost my train of thought there, I was zoning out. There is such a thing as a polypeptide. A polypeptide is when you join a chain of amino acids. Now, a protein could have 50 to 2,000 amino acids in the chain. Think of it like prayer beads or something like that on a string. Like you have your prayer beads on a string. Each one is an amino acid. So you could have up to 2,000 in certain proteins, or you could just have 50 in the smallest proteins. Depends on their job. Things like viruses, which are very small, have very simple proteins with only a few amino acids, or 50 or 60 amino acids. This is the reaction of some amino acids. I've got 2 amino acids. Notice I've just put an R group here because that's the side chain. They could be any length, the different amino acids. Now, condensation means that we're going to lose like the OH here and an H here. Let's get rid of that. That looks a bit dodgy, that. So condensation is when a water molecule breaks off. And it's exactly the same. Condensation reactions and hydrolysis reactions are exactly the same in for lipids and for all different molecules, carbohydrates, for all biological molecules. We form larger molecules by condensation, and we break larger ones up in digestion by hydrolysis, hence the need for water for digestion. As you can look over here, you can see a water molecule is missing, and the peptide bond you've got over here is your peptide bond. So over here we've got a peptide bond. Oh, I think I'm missing a hydrogen on my amino. Am I missing a hydrogen there? Hold on. No, I'm not. I'm all right. No, we're all good. I'm not missing a hydrogen. Okay, yeah, so this would be like a peptide bond. And that's formed between two amino acids. And we call that a dipeptide. You could have a tripeptide if you had 3 amino acids. And then, of course, you can have polypeptides if you have really long amino acids. This is a peptide bond. So you've got your 3 bonds. Yeah, you're right. You're right. I should put H2O over here. Absolutely. Yep. And again, if I was doing this as a polymer, if this was A-level chemistry year 2, A-level chemistry, I would need to open the ends of the carboxyl group, the COOH, need to get rid of an OH over there, and I need to get rid of a 1H on the nitrogen and show the ends open and put brackets there and put an N for as many as possible. And again, I could do the same with the water molecule, put an N next to it, because you'd need a, yeah, basically to show that it could go on and on and on. But in bio, you won't have to do that. Yeah, and we would call this, you know, I could label this, I don't have enough room, I don't think, but condensation going this way. This way is condensation. And if we go back the other way, it's hydrolysis, like that. Going back the other way. And then this is the A-level bit. As I said, it's just a quick lesson today. It won't take more than 40 minutes. It's my prediction. I'm not usually right with the predictions, but today it should be no way more than 40 minutes. So proteins have 4 structural levels. So this is a bit of A-level new stuff as well. Proteins have 4 levels of structure. We've got something called the primary structure. Now, do you remember where proteins are made? Proteins are made on the ribosome. Now, if you did triple, do you remember the process where proteins are made? There's a name for protein synthesis. A couple of keywords for protein synthesis. They're actually made on a ribosome. You might remember the name of that process. It begins with a T. When we make a protein, it involves transcription, where the DNA is read inside the nucleus, and then it involves translation on a ribosome. Yeah, that's right, Karam. Yeah, that's right. Anyway, when we finish with translation, which happens later, happens after transcription, it happens on the ribosome, and all the tRNA molecules have moved off, and the mRNA's gone off to be digested. We're left with different amino acids. These squares and circles represent amino acids and these weird shapes. Obviously 20 different shapes, and proteins would normally have 50 amino acids, but that would take me too long to draw them all out. So if we imagine these are different amino acids together in a chain, they make a protein. Now the specific order is known as the primary structure. So what we say is that the specific order of the amino acids is called, how do I highlight that bit there, that box? Probably like that. No. Oh my good gosh. No A-levels going on. No A-levels going on. I don't know why this text box keeps coming up. I'm just trying to click on this box. Oh my gosh, that took me long enough. Had a brain fade today. So this is the definition of a primary structure. Primary structure is the sequence of amino acids in the chain, in the polypeptide chain. The bonding between the amino acids is peptide bonding. So you may be asked what type of bonding is in the primary structure of a protein. So that protein is no good to anyone in a straight line like that. What do you think's got to happen to that protein chain for it to be able to do a job? If it wants to be an enzyme, what's it got to do? What's special about enzymes? What special regions do they have? Do you remember? No, Rizwan, this is just, I'm doing Introduction to Biology A-level. So, yeah, I've already done 2 lessons. Yeah, that's brilliant. They've got a specific active site. So as a straight line like that, if you imagine again, like some prayer beads in a row like that, with all the beads along a piece of string, like that, it's kind of an active site. So we need to fold it and bend it round. And so the primary structure is the order of the different amino acids. And by the way, it says here, you can make 10 to the 390 different protein chains. So that's huge. That's 390 zeros, 10, and I don't even know the size of that number. You can't even put a name to, I don't know what name that number would have. It's so huge. It's bigger than Avogadro's constant. It's absolutely huge. So there are infinite almost amounts of proteins you can make. So they then have a secondary structure. What that means is proteins, after they leave the ribosome as a straight chain, immediately, because they have different R groups, the proteins start to fold into different shapes. And there's 2 main shapes that all proteins fold into. And one is known as an alpha helix. You remember the double helix in DNA? This is a single coil like a spring. It kind of coils round like it coils round like a spring like that. Or a beta pleated sheet, which is kind of like it. If you just took paper and you folded paper, yeah, I got a D in art for GCSE. If you folded paper, that would be a beta pleated sheet like that. So, and what makes them, how do they know which one to go into, which shape? Well, they don't. It depends on the hydrogen bonding that forms between the different amino acids. So we have something that you'll learn about later called hydrogen bonding, and it's a kind of intermolecular force. So it's a weak force between different amino acids. It's not quite a bond. A peptide bond is covalent. So is a glycosidic bond. So is an ester bond. All the strong bonds involved in biological molecules are covalent bonds from chemistry. But this hydrogen bonding causes the straight chain of the amino acids to immediately fold after they've left the ribosome. And it's hydrogen bonds that form between the amino acids that cause this secondary structure. So you can be a beta pleated sheet, or you can be an alpha coil or alpha helix. Right. This is a proper drawing of them off the internet. So that's just like, well, another diagram, just so you can see the alpha helix, the chain curls around itself like a coil, as the beta pleated sheet is like that. So they're 2 different structures, and all you really need to know about them is that the bonding that causes that is called hydrogen bonding. Now there's a tertiary structure because there's 4 levels of protein structure. Once they've formed that level of bonding, they then further fold into a 3-dimensional shape. So you just got to think of a 3-dimensional shape like this cup. So like they might have an active site. So they now fold further into a 3-dimensional shape, and we call this the tertiary structure. And what causes the tertiary structure is that when the hydrogen bonds cause them to fold, ionic bonds started to form between positive and negative groups on the amino acid. So wherever there's a plus and a minus, we started to get ionic bonds form. We also have something called hydrophilic interactions and hydrophobic interactions, which there's water-loving parts of the protein and water-hating parts, and they start bonding with each other. And there's even something called disulfide bridges, which are covalent bonds that form between things called cysteine groups. Cysteine groups are R groups that contain sulfur atoms. So occasionally, some amino acids have sulfur atoms in them on their R side chain. The use of alpha and beta, they're just two different ways of folding. So how does it affect the overall structure of the protein? Not much. They then will go into a tertiary structure anyway. But if you were to look closely, you would see that it's got beta pleated sheets or an alpha coil, but it's going to further fold anyway. So it just has a secondary structure there that then it carries on folding into a tertiary structure. So it doesn't really massively affect it. I haven't looked into whether certain particular types of protein have beta pleated and certain types of protein have alpha helix. That's probably whether the ones with the beta pleated sheet are structural and therefore not soluble, are insoluble, and the alpha ones with the alpha coil are soluble. That might be the case, but that's like beyond the specification. So now for proteins that are just made of one polypeptide chain, this is called the final three-dimensional shape, the tertiary structure. It's known as the final three-dimensional shape. And what that means is it's very important because this is what creates the active site on an enzyme. You remember that's where the substrate fits with the lock and key hypothesis, or the induced fit. The substrate fits into the active site. So it's very important that the folding happens correctly. And all proteins are specific, not just enzymes. Remember that if you caught COVID, your body would have to make antibodies, which are proteins, with a specific shape that could lock on to the antigen of the COVID to destroy it. So the folding and the bonding is very important. That's how they change the kind of specific shapes. Now, what would happen if we changed the original primary structure? If we took one of the amino acids out, well, that would affect whether it's going to be a beta pleated or an alpha coil, and it also might affect the tertiary structure, the final 3D shape, and mean the enzyme can no longer do its job. So when people have a genetic mutation, which we get all the time, most of them are pretty harmless because the change of one amino acid might not change the whole structure. But if you do have a genetic mutation, say, that means you can't produce an enzyme for something, then yeah, that could be deadly, or it could give you an inherited disease. It could give you a disease. So for people that have, yeah, well, really, this is basically A-level. Yeah, this is the third lesson I've done in the series. The last 2 weeks, I've been doing some A-level biology lessons. And so this is the third lesson in the first topic of A-level biology, which is biological molecules. So you can get to see, if you're year 11, you can get to see what, you can get a look at like what A-level biology is like, the kind of level of detail and difficulty. And if you're doing A-level, this is actually the content you'll need to know for the A-level. Obviously, it can be misleading because this topic, biological molecules, has got a little bit of chemistry in it, like as in bonding and elements. And a lot of biology is not about that. And this is quite a content-heavy part, whereas other parts of biology are more concept-heavy, where you've got to understand slightly more difficult concepts. Doing them for chemistry. Chemistry was yesterday. So I did the second chemistry one. I'm doing chemistry every Monday at 8 and biology every Tuesday at 8. Chemistry yesterday was the electronic structure. Now, I hadn't got my mic on for the first, it was an hour and a half lesson, but I didn't have the mic on for the first 5 minutes. So a bit of a disaster there. I might redo that one at some point, but it was just basically talking about the new way of doing the electron shells. So you can have a look at that. And there's a week before that, I did one on mass spectrometry and the history of the atom for chemistry. I'm not doing any on maths. I'm not a maths specialist. Like I can do maths, but maths A-level is, yeah. I've done biology for 25 years and chemistry for 25 years. So like, I know those courses. It doesn't mean I still don't make mistakes. mess something up occasionally. Yeah, I messed up one question in the GCSE lives I did, but I corrected it. But yeah, there we go. Yeah. So you can watch it if you're year 11, or you can watch it if you're going into a team SIAR. Are you going into year 12 in September? Or are you in year 11 in September? Yeah, okay, cool. Yeah, so this will give you an idea. You might still be thinking, do I take chemistry? Do I take biology? Do I do physics? Do I do math? This might help clear up or make you aware of like the kind of stuff. I mean, chemistry, it's normal to be baffled. So chemistry, I would say conceptually chemistry is harder. Biology is much more content heavy. But biology has the lower grade boundaries because people find the exams harder. Right. We've got the quaternary structure. Give me 1 second. Oh. This is the fourth level. The fourth, and we're nearly finished with proteins now. Proteins is okay. I've just noticed something. Spelling mistake, spelling alert. There's certain things that your brain, certain words that you might find hard, that you might find weird to spell. And saying quaternary structure when it's supposed to be quaternary structure is a mistake I've made so many times. Say quaternary structure, it's blooming quaternary. I still had to Google it while I'm sitting here talking to you guys, which is a bit of a nightmare. Yeah, no probs, no probs. What you can see hopefully here is that some proteins don't just have one chain, they have 2, 3 chains. So imagine you had like, I don't know, a red set of prayer breeds, a green set and a blue set, and you started wrapping them. Not only were one of them wrapped up into a shape, a 3-dimensional shape, but you now weave another one in between and wrap it around, and then a third or a fourth. And we call that a protein that has a 4th level of structure, a quaternary structure. And that's when you have more than one polypeptide. And in that situation, that becomes the final 3-dimensional shape for the protein. And some examples of proteins with a quaternary structure, just notice that spelling mistake again, are collagen, hemoglobin, and insulin. Collagen's in your skin. So it's collagen. As you get older, you don't produce the protein collagen as much. So, yeah, you get wrinkles. So when you start getting wrinkles in your forehead and stuff, that's your body is not producing enough collagen. And it's what kind of makes your skin flexible and smooth. And as you get older, you start messing up your production of collagen. And some people have collagen injections or collagen fillers in their face to get rid of wrinkles, or they have them in their lips to puff their lips up. Seems to be a thing nowadays, everyone mutilating their bodies. But collagen's a protein. Hemoglobin, do you remember what hemoglobin does? Hemoglobin is an important protein. It carries oxygen around the body. It actually binds with the oxygen, has a very specific 3-dimensional shape which allows it to bind to oxygen. Yeah, absolutely spot on. Binds to the oxygen. So its 3D shape is really important. If you change a couple of amino acids in one of its chains, its 3-dimensional shape changes, can't pick up oxygen and you're going to die. So there are certain mutations which would be fatal. And maybe other people have mutations which alter the shape of their hemoglobin and make it better at picking up oxygen. So there are all sorts of things. Insulin, you remember insulin. Well, insulin is a hormone that's involved in the control of blood glucose. Why does that need a specific 3-dimensional shape? Do you remember what insulin? Yes, hemoglobin has iron in it. So it's a really good point, Karam. And later on in next week's lesson, I think I'll be mentioning that because that's part of next week's lesson. Because proteins aren't done with. Yeah, so you'll find like within a hemoglobin molecule, you'll find an ion, Fe2 plus or 3 plus, can't remember which one, Fe2 plus. You'll find it, 4 of them are in a protein. So each hemoglobin molecule, which is made of 4 polypeptides, this only has 3, a blue, a green, and an orange. But there are 4 chains in a hemoglobin, and each one has an ion within it that can bind to an oxygen. So, yeah, hemoglobin can pick up 4 oxygen molecules because of the ions that are in there. Oh yeah, nice. Really good point, Karen. However, we would say that the ion, and that's a really smart thinking by you, we would say that the ion is not part of the protein. It's not bonded to the protein. It's like trapped. If you imagine the polypeptides as like a cage and the ion is in there, so it's not actually part of the protein. And we'll come on to, there's other ones like chloride ions attached to some proteins like amylase, which is an enzyme which doesn't work without the presence of chloride ions. There are groups that are held within proteins. They're called like prosthetic groups and cofactors and coenzymes. There's all these other things that work with proteins, but they're not part of the actual chain. So yeah, you won't find Fe atoms in an amino acid or even on the R group. So they're just held loosely within the structure, or they might not be loosely held, but they're within the structure. But they're not part of the chain. Good question. And insulin, which I was talking about earlier, has to bind to liver cells and muscle cells, and there are specific receptors, and it has a specific shape, and it binds, and that causes channels to open and glucose to be taken in. Now, if you've become insulin resistant, then probably the shape of your insulin is no longer connecting and your cells are no longer responding to it. So it may be that the shape has been altered. So if you alter the shape of any of these proteins, and that starts with the primary structure, if you change the primary structure, it's going to affect the final 3-dimensional shape, the tertiary structure or quaternary structure, and that's going to affect the job of the protein. Okay, yeah, enzymes are proteins, we know that. Just down here, it just a little bit more on enzymes. It just says they're roughly circular and they're soluble. If you're doing, it depends if you're doing AQA or OCR as to whether you need to know extra stuff, but soluble proteins are known as globular proteins, and enzymes have to be soluble. They're moving around in cytoplasm, they're moving around in liquids in the body, so they need to be soluble. However, the proteins in your fingernails and in your hair and in your skin, you don't want them to dissolve when it rains, so they're insoluble, and they're known as, they're structural proteins and they're insoluble. Because we've got globular and structural. Antibodies are proteins that you need a globular as well. They need to be moving around the body. We're nearly finished for today. Antibodies are made of 4 polypeptides. They have 2 light chains, 2 heavy chains, and they've got a variable region, a region that varies between each antibody so that they can be specific to the antigen on a pathogen and bind to it. Now, you're going to do a load more in a topic on immunity in A-level biology, where they really go into antibodies in a lot more detail. And then these are just some other examples of proteins. It's all very brief in this initial topic. I'm just following the syllabus through, and it's all brief at this stage. And some of these brief topics, they go into a lot more detail just in a few units time. So we have transport proteins that are found in our cell membranes, and they have hydrophilic and hydrophobic parts. Hydrophilic means water loving, hydrophobic means water hating, and they fold up in a way where the hydrophobic parts are on the inside and hydrophilic on the outside, and they make a channel that allows certain molecules like glucose to enter into through cell membranes, because cell membranes are designed not to let a lot of things through, especially water soluble things. Cell membranes are made mainly from fats, from lipids, which we did last lesson, and there's a whole unit on cell membranes, but the cell membrane is designed not to let water soluble things through, like salts, amino acids, glucose, and all these things. So they need special channels, pathways to enter the cell, which are created, and that gives the cell control over what goes in and out. And structural proteins, I was talking about your fingernails, your hair. Your fingernails and hair are made of keratin, and that doesn't dissolve. And then your skin is like collagen, which keeps your skin flexible and smooth. Now, structural proteins are a little bit like cellulose, which was a carbohydrate we looked at in the first biology lesson. They have cross links between the layers, a bit like plastics and polymers when you talk about thermosetting and thermohardening or softening polymers and things. Basically, they have lots of cross links that make them more rigid, insoluble, and tough, so they're for structure. Okay, you do need to know the test for proteins, the final bit of information on proteins for this lesson. So, and then just 2 exam questions and then we're done. So, do you remember the test for protein from GCSE? You might remember the test. And you might remember the colour change. Yeah, awesome, Karam. Well done. It's the Biuret's test. He says, I'm flicking through the book right now just to see what bonus stuff you need to know. Okay, and do you remember the colour change? Yeah, brilliant. Blue to purple, pink, or lilac. Nice. Yeah, you can say purple, pink, or lilac. What's cool is that as a memory recall way, you already remembered it, but as a memory recall way, you can kind of think of like the P, purple or pink with protein. You can kind of think of that. And you can kind of think of the B, Biuret's, a bit like Benedict's begins with a B. It starts out blue and then it turns pink for protein. So there are some sort of, if you're someone who struggles with memorizing little bits of content like that. What else do you need to know? You actually need to know what Biuret's solution is made of. So all you need to say is add some sodium hydroxide. Do you remember what sodium hydroxide is? You might remember what sodium hydroxide is. It's also in Benedict's reagent. Yeah, very good. Very similar to Benedict's reagent. It's copper 2 sulfate. The Roman numerals tell you here. I've just done 2 letter I's, but the Roman numerals, then it will turn from blue. Oh my gosh. Yeah, the Roman numerals, do you remember what they stand for? They tell you something about the copper. Cool. That's right. So it's a copper 2 plus ion. So with metals, especially transition metals, they have, well, they don't do it with other metals because you just look at the group number. But with copper, which doesn't have a group number, or iron or nickel or chromium, they have to give you the Roman numerals so that you know what charge is on the ion. Cool. Okay, just a couple of questions before we finish. Nice and easy lesson. So a dipeptide consists of 2 amino acids joined by a peptide bond. Dipeptides may differ in the type of amino acids they contain. So there's 20 different ones. So there's loads. I don't know what the combination that is. I don't know how you do the maths. You see, that's why I'm not teaching maths A level. But if you've got 20 amino acids and they've got to pair up, how many different combinations are there? Jeez, I don't know, but it's a lot. So describe 2 other ways in which all dipeptides are similar and 1 way in which they might differ. So if you think about amino acids, what's 1 way that all amino acids are similar to each other? Or 2 ways that amino acids are all similar to each other? What groups do you find on all amino acids? And what group, and what's a different part of an amino acid or something that's different? Nice, you're all over it. Same amine, same carboxylic group. Some have different R group. Nice. So that's going to smash it, really. You've got it there. So they all have same amino group or amines. They contain amines, contain carboxylic group. You can say carboxylic acid if you like. So that's going to cover all of those points. All contain the elements. What did we say the elements were? Ah, yes, sulfur. Nice. Yeah, so carbon, hydrogen, oxygen, and nitrogen. But the difference is some have sulfur. Really good. Sulfur atoms. And then different R groups, which kind of covers sulfur atoms as well. We could just say different R groups. Yeah, perfect. Well done, you're on fire. So as always, yeah, so these are the answers from the mark scheme from AQA for that particular question, which was I think 2017 or 18. I was scouring through their exam papers that are published online on their website. So similarities, amine groups at the end, carboxyl groups or COOH, all contain CH, N and O. The difference, they'll have different R groups. Yeah. Cool. Another question. So let's read through this one. Scientists measured the mean amino acid concentration in white wines made from grapes grown organically and white wines made from grapes that were not grown organically. Which test could the scientists have used to identify are there amino acids in the white wine? Okay, so they just want to find out, and you've already named this one, so you know the answer to this, guys. The test for amino acid, Buretts. Yikes. Buretts or Buretts, however people want to say it. Draw a box around the part of the molecule that's the same in all amino acids. So which bit of this is the same in all amino acids? So what we've got to do here is identify our carboxyl group, identify our amine group, and then go, aha, I need to circle all of that, because all of that, let's just, now that I've identified those 2 groups, let's just circle the constant region. So the bit that's always the same is this bit. And this amino acid is isoleucine, one of the 20 amino acids. And up here, you can see here is the variable region, the R group. So this is known as the R group, and that's the bit that can be different on different amino acids. And in chemistry, A-level as well, they like to use R for chains which can be completely different on something that's the same, just to save hassle. Name the chemical element found in all amino acids that's not found in any triglycerides. Which chemical element is found in all amino acids, but not in fats, lipids, triglycerides? So it's found in all amino acids. Yay, nitrogen. Well done, well done, well done. So nice one. Thank you for that. So round of applause. Yeah, so little round of applause. Well done. Listen, awesome. Gonna finish. A big shout out to Karen, by the way. Karen turning up and getting involved in the lessons. Awesome. And obviously, you know lots of the answers at this stage. You're on fire, so keep it up. That's good, looking good for you for when you choose your A levels, whatever ones you do. So you're doing really, really well. So nice one. And yeah, shout out to everybody else who joined the lesson. So keep it up. No props. You're looking good to be really successful in your A levels. So yeah, I'll be back next week with chemistry and biology. So yeah, just tune in. It's going to get a little bit more difficult after a little while when we start going into the harder topics. So next week I'm going to be doing ionization energy for chemistry, which provides the evidence for those orbital structures and the electronic configuration. So it provides the evidence for that, and it explains a few other things. We're going to look at how ionization energy changes across a period and down a group, and what actually ionization energy is. So that's what we're doing in chemistry next week. And for biology, it's a little bit more on proteins. And then, yeah, that's it. All right. Take care, and thanks for tuning in. I'm off.